Our last finalist to present is MeMed, represented by their CEO, Aldin. To meet the urgent need of risk stratifying SARS-CoV-2 positive patients, MeMed is developing MeMed COVID-19 Severity, an easily interpretable host protein signature that includes virally induced proteins. Dr. Aldin, please tell us more about this new technology.
Okay, so good afternoon. Hi, my name is Aaron. First of all, I'd like to thank the organizers for the kind invitation. I'm one of the co-founders and CEO of MeMed, trained as a computer scientist and molecular biologist at the Weizmann Institute and the Technion, the Israel Institute of Technology. I was on my way to academia when I met this guy, Dr. Kfir Oved. In his grandmother's kitchen, we started to work on this crazy idea, and that crazy idea grew to a company beyond our wildest dreams. We took it from inception to our recently, I say recently, last week, landmark FDA clearance. Today I'll share with you our story. Yeah, it's working, okay. So we're based in Haifa, Israel, headquartered, and also growing right now a base in Boston, raised well over $100 million in equity and another $33 million from the US Department of Defense and the European Commission. And what we do at MeMed is based on a simple idea. Our body's immune systems have evolved to tell us what's going on inside. And what we do is listen, develop technologies that can potentially transform the way that we manage patients with acute infections and inflammatory disorders. It starts always with a patient, not with the technology, with the patient. So imagine you go to the doc with your child with a fever, snuffles, or maybe it's your parents, a seemingly simple problem that gives rise to some of the biggest healthcare challenges of our time. We'll see in a second. There's several clinical dilemmas that come to mind. The first one, is it a bacterial or viral infection? Do I treat or not treat with antibiotics? Now, 1 in every 2 antibiotics is erroneously prescribed to non-bacterial infections because bacterial and viral infections are clinically indistinguishable. 1 in 5 patients are supposed to receive antibiotics are not receiving. That has a lot of consequences. In total, a very big both health and health economic consequences, including the rise of resistant strains of bacteria, one of the biggest healthcare challenges of our time. The second dilemma, the same patient will have a severe versus non-severe outcome. So 50% of all hospital admissions or deaths are due to severe infections. Time matters. The earlier we can catch it, it can save lives and reduce costs. So obviously, we have many conventional tests out there, from cultures, PCRs, rapid antigen tests, and they're good, and they're helping push the field forward, but there's several gaps that still remain. The first, inaccessible infection sites. So you can use a multiplex PCR in cases of pharyngitis, but what about otitis media? What about sinusitis, bronchitis, pneumonia? What about a child with a fever without source? He's just screaming in daddy's and mommy's ears, and you don't really know whether to take a nasopharyngeal sample, a urine sample, a stool sample. Try, by the way, to take a urine sample from a 2-year-old girl, like the one that I have. Very, very difficult. 1 in 4 patients have this. Number 2, often no pathogen is detected. I'm talking about even if you use a multiplex PCR or a gene expert or a biofire, 50% of the cases, nothing's going to be detected. Third, false alarms due to colonization. You identify, say, a rhinovirus in the nasopharynx of a 2-year-old child. What does that mean? Every second child, whether healthy or sick, will have a rhinovirus. And lastly, poor performance due to emerging strains of pathogens. Now we have SARS-CoV-2, before that H1N1 swine flu, then MERS and SARS and what have you. So these are challenges, real big challenges. So the vision behind MeMed. When we set on this journey, we imagined this small benchtop device, a few minutes, small volume of blood that would somehow decode fever. Solve these 2 clinical questions, bacterial versus viral infection to treat with antibiotics or chicken soup, and severe versus non-severe, do I escalate treatment or not? Now, the paradigm is straightforward. Real-time quantification of the body's immune system coupled with machine learning, we create like this barcode of the immune response, sensors, algorithms, bacterial, viral infection to treat, not to treat, and the same concept for what's called a MeMed Universal Severity. That's the second product. Now, ideas are cheap. Making technologies harder making products even harder, getting cleared and manufactured even much harder. So what I'll try to do right now is take you through this decade-long journey and compress this into maybe 2 minutes. 4 years discovery and and research, 5 years of product development, 7 years of clinical validation, EU cleared, and last week FDA clearance. So we started, again, 4 years with the largest proteomic ever to be conducted of human response to acute infections, 100,000 potential protein isoforms, bioinformatically down-selected 600 proteins, and then eventually came up with 3 proteins. The first study, again, there's obviously some machine learning behind them, compressing this to a single number. The first study published with a performance sensitivity and specificity of over 92, 93%. It was accepted as a potential breakthrough, but a lot of skepticism. People are telling us you need external, not internal, prospective, double-blind studies led by multiple doctors around the globe, published in the top journals. Very skeptical, for a good reason. The field has been plagued with overpromise and underdelivery. So we took the technology and gave it to multiple doctors around the globe. We told them, whatever comes up, publish. If it kills the company, none of the people at MeMed needs job security. So they ran this for multiple years. That's what you're seeing here. Today we have data on over 20,000 patients, prospective double-blind studies and real-world evidence. Some publications in The Lancet and up to date. It's working, okay. Over 20 studies, 30 medical centers, and as I said, over 20,000 enrolled patients. Okay, the second challenge. So everything we talked about, again, cool technology, but it doesn't mean anything if you can't really fit this into the workflow. Now the challenge is some of the proteins are picogram per ml, some are nanogram per ml, and some are microgram per ml, and we need a very tight CV. How do you do that? We tried to use some off-the-shelf technologies, lateral flow. We failed miserably and spent a lot of dollars for about 2.5, 3 years. And then, again, this is just a caricature. We tried to somehow explain to people what we're doing. So, right, picogram per ml is like having a soccer field, 2 kilometers of M&Ms, running a random number between 0 and 1,000 Skittles. You have to go and count the Skittles. You have 15 minutes, go. That's the challenge. Yeah, well, we didn't laugh, by the way, I can tell you, when it didn't work. So fast forward, the idea here, this was an idea conceived by Dr. Kfirovid. The idea was, let's take a huge machine, an Alinity, a Telica, Liaison. It can actually do picogram per ml with single-digit CV. It actually works. We know that biochemistry works, but it's very costly. So let's miniaturize that. It's an engineering problem, not a biochemistry problem, which is much harder. And again, 6, 7 years into the process, came up with MeMed Key. This was developed, folks, around 4 continents. You plug the cartridge inside, put 100 microliters of serum, press the button, 15 minutes, and that's it. Boring. All the complexities masked. You get a quantitative interpretation and a qualitative interpretation, score between 0 and 100. The higher the score, the higher the likelihood of bacterial infection. Mixed infection, receive a high score. It can measure multiple proteins, rapid results, designed to be clear-wearable, and it can basically measure any protein in your body from oncology, metabolic diseases, wellness, and veterinarian. Okay, moving to the next. Universal severity. We worked on that, and then this little virus hit us. COVID severity, or COVID pandemic, created an urgent need for an accurate and easy-to-use tool to predict the disease severity, but 35% report uncertainty. Clinicians, 20% over-admission and 8% under-admission. It's a huge dilemma, huge problem today. Now, the challenge, immune response to COVID-19 is highly complex, transcriptomic, proteomic, cellular level, metabolomic. So we came up with 3 proteins. One is called TRAIL. TRAIL is the world's first viral-induced protein that's used as part of routine care and is cleared by FDA. It goes up in viruses, and it's almost disappearing when you have a severe outcome. Second, IP10 is an infectious biomarker and goes up when you have a lung injury. And third, good old C-reactive protein. It's a general hyperinflammatory severity marker that's been around for about 40 years. Combine them together, machine learning, and you get a score. Score between 0 and 100. The higher the score, the higher the likelihood of a severe outcome. Severe outcome defined as ICU level of care or mortality. I have to really press, by the way, very strong on the button. I don't know if you've seen it. Really trying. Okay. So here's a real clinical case. Again, it's about the clinical case, not just the technology. 65-year-old male, Clinically stable, saturation 94, presented with a cough. What do you do at the ED? Sent home, admit. Say you have a score of 4, that's a very low risk. So you can potentially send home without the risk of deterioration due to care not received. 86, consider admission. So that's just an example of how you can use this technology. So we have this slide that talked about regulatory status, CIVD, and explanation where we are with FDA. But then we just heard back from FDA and just announced this last week. So FDA has now cleared the first technology to aid in distinguishing between bacterial and viral infection based on the body's immune response readout plus machine learning. It's based on a 5-year intense interaction with FDA. We're grateful for that. We've cleared both the signature and the platform. It's based on yet another multi-center prospective double-blind studies of over 1,000 patients, some of these leading medical centers in the US. We had to overcome this huge challenge of how do you come up with a reference standard in the absence of a real objective measure? How do you actually say what your performance is? So we had to work with FDA on this for many, many years, and we passed both the primary and secondary endpoints with flying colors. We almost lost a few folks also due to a heart attack when we opened the books, but they survived. So we got a very broad indication for use. FDA has cleared this for both children and adults. Suspected acute infection, lower respiratory tract infection, upper respiratory tract infection, urinary tract infection, systemic infections, EDs, and upon admission, but also urgent cares, which is really important. COVID severity, okay, regulatory. We got it CIVD cleared, and right now we're in very intense dialogue with FDA. Moving on to go-to-market strategy. We have a unified host response product pipeline positioning. You have a lot of products. How do you leverage them together? So we're leveraging the same core technology, same call points, same install base, same KOL network, and these are complementary market dynamics. BV is growing. AMR was there before COVID, exacerbated during COVID, and it's going to become even a bigger problem tomorrow. So it's stable. COVID, obviously volatile, and then universal severity, also stable. So we have using both volatile market dynamics, but also things that are steady. Business model, 2 parts, direct and strategic partnerships. Razor and razor blade model, plus we collect the data. We go direct in the US and Israel, plus a few countries in UK, other places distributors. We start in the hospitals and EDs, then IDNs and urgent cares, and then physicians' offices and retail clinics. So there's a lot of thought behind why that is. But we can't really take this, here again, will it work? Yes. We can't really take the market by yourself. It's just too big. Child with fever without source, snuffles, LRTI. So we partnered, more specifically, Fred and the team selected very specific partners. The first one, Diasorin, that has a large automated immunoassay machines covering about 8,500 hospitals in Europe and the US. To complement what we're doing, here you see a quote by Carlo Rosa, their CEO. I think they believe this is a strategic project for them. Market. The market is big. This is our FDA indication for use, about 250 million cases. Even if you break it down to different segments, just the top upper echelons, is about $5.3 billion market opportunity. And for COVID severity, obviously the market is much more volatile. It's between 200 and $1 billion market opportunity. Pricing. Well, gone through a lot of work and doing surveys on hundreds of stakeholders from pediatricians, internists, nurses, CFOs, laboratory managers to come up with a model that we think will allow broad accessibility. Reimbursement, going through the playbook, PLA, then CPT. Hospitals, it's a blended average, obviously DRG, APC, and fee for service. You need to show health economics. As you move to urgent cares in decentralized setting, you need your own CPT code, and there the name of the game is clinical utility, and clinical utility. And we've been working on that as well. Okay, competitive landscape. So MeMed is a funny company. So we have both, we're a host response company, but we also have a platform, both of which have a lot of independent utility. It's almost like 2 companies combined together. Now, I'm going to focus only on the competitive landscape on the host response. The other one is a different story. Okay, so first of all, how does this compare to conventional direct pathogen detection tools? So for example, multiplex PCRs. Let's talk about our disadvantage. Unlike a BioFire, we cannot detect the substrain of pathogen. Is that important? Yes, it is. For some patients, especially the centralized setting, but as you move from the centralized to EDs and decentralized, sometimes it's not actionable information. It's all about not technology, but actionable information. What is the advantage of using this paradigm, new paradigm of host response? It's not just a geeky nuance. It allows you to diagnose inaccessible infections because the immune system circulates throughout the entire body. It allows you to not be affected by the natural flora. If that rhinovirus is not attacking the body does not counterattack. So we solved that problem. We didn't do that. The immune system did. It's robust to evolving strains of bacteria. We've been able to show that, and we can do this within minutes. What about other biomarkers? So there's many things out there, but probably the prominent biomarker, the only one that's cleared by FDA for antimicrobial stewardship is PCT, procalcitonin, and then for COVID severity, it works, okay. COVID severity, interleukin-6, EUA. Now, there's additional promising tests there right now that are under development in host response, but they've not yet been FDA cleared and still lacking prospective double-blind large studies, so the jury's still out there. I want to focus for a second about some of those differences between these competing things. So obviously there's pros and cons for everything. We think we have a unique positioning, particularly if you look at the accuracy, which is important. At the end of the day, we're treating here patients, children, adults. So here we see a independent, again, comparison between the signature, this is a raw curve, area under series operating curve, of the signature compared to, say, what blood count, which is routinely used, monocytes, A and C, and procalcitonin. It's not a little bit better. It's a game changer. Here you see, here you see, I ran too fast. Trying to go back, okay, it's not going back, but what you would have seen in the chart is a comparison between the MeMed COVID severity, yeah, now it is. It's working. Interleukin-6, ferritin, and PCT. Now it has a life of its own. Okay, I'm just going to put it like this. I'm not going to touch it. IP. We have over 100 patents and patent applications, many of which have already been granted, both on the signature, the biomarkers, but also the suboptimal signatures. So we have hundreds, thousands of different configurations on the reagents, on some of the elements of algorithms, the device itself. Freedom to operate, solid position, and also not easy to design around. A deep product pipeline, what would you do if you had this white canvas? Both the platform and this biobank that we have and these capabilities, what else would you do? So here's a list of some of the stuff that we're going after, something called cytokine profile or MeMed X. Hopefully in one of the next AACCs we're going to be able to share on that. The team, probably the most important thing that we have is the team, and here's just a partial representation from here in the audience. I think I can see here, I can see him there. So Troy recently joined, former head of commercial microbiology for Thermo Fisher. Fred, former corp dev at bioMérieux. Moran Barak, former VP medical Novartis, Takeda locally. Oren, CTO of Mazor Robotics. Jim, head of sales for BioFire. Tanya from Cambridge. Chris Hibbert from Biosys, a very round team. And obviously multiple partners, both investors and medical centers from around the globe. To summarize, it starts with a simple idea. Our immune system has evolved to tell us what's going on in our bodies. And we're developing technologies that can potentially transform the way that we manage patients with acute infections and inflammatory disorders. We took it from an idea in grandmother's kitchen through technology, product, manufacturing, regulation. And through this journey, we believe now that this novel field, novel category called host response has finally come to this time that it's prime. It's ready to start having real impact on real patients. Thanks.
All right, panel, it's all yours.
Thank you. Maybe I can go ahead. Good presentation, Aaron. And congratulations on the FDA approval and over 130 million in cash raised. That's amazing. So that definitely validates the sophistication of your platform. My question to you is, you were mentioning workflows and problems you're trying to solve. How about the behavioral problem of this triage doctor? You know, how are you convincing them that they change, you're disrupting their standard of care? Now they have to add an additional step, maybe draw blood, get additional information. Is there an element of reduction of hospital errors, or is your angle insurance? How are you ensuring faster adoption?
Okay.
It's a loaded question.
Absolutely. So where to start? You're right. Sometimes changing behavior is harder than developing technologies. And just came back from a trip actually in Phoenix this week, or last week actually, talking with multiple clinicians. So here's the thing. You start where it really hurts, where you actually don't need to change the workflow, because good luck. If you want to change the workflow, good luck. That's really, really hard. So what about, let's start with a real clinical case, a real clinical dilemma. A child with a fever without source. So the current workflow is as follows. They come to the doctor, maybe it's an ED or physician office. Mom and daddy are hysterical. They want something. You usually take blood anyhow, because it's part of the workflow, CBC, what have you. And then you have to basically try to figure out, where's the infection site? Do you take a nasopharyngeal sample? Do you take a urine sample? Or do you take a stool sample? And you don't really know. And by the way, good luck trying to take a urine sample from a 2-year-old girl. Good luck. With catheter and maybe a diaper and what have you. So a lot of money, a lot of anxiety, maybe LP. If you miss it, it's just a big problem. Overtreating is also a problem. Antibiotic, many times IV, and you have to hospitalize. Parents have to stay as well. So starting there, if you talk with the clinicians, they actually get it. You don't go and try to boil the ocean. Here's, I think if you can put the slides, I can see the slide. I don't know if you can see, if you can just stick back to the slide. Okay, so this was hidden there. So this is, once FDA clearance came, these are just quotes from folks that are using the tool right now. So, okay, this is, Rich Bacher from Harvard and head of Division of Emergency Medicine. They get it. He says, well, for over a few decades, we've been waiting for something like this. It's extremely hard. This is Dr. Adi Klein. She's the current head of the Pediatric Society of Israel. So she's saying, introducing MeMed technology has made a significant impact on our medical practice. And she's giving an example, not of the 100,000 patients, and one patient that it really helped her, not by the way, only case reduced overuse, she actually caught an underuse. That actually occurs more than what you would think. So you start there, and then it starts to spread. Same goes for, by the way, adults with LRTI, which is another big problem. And we're working with them together. Obviously, it takes time.
Okay. Thank you.
If I may, I could actually pick up from 2 different points. I'll follow up on the fever of unknown origin. So it comes back to all my questions around specificity. Again, you've got, let's say, bacterial versus viral, but you still don't know the source. Yes, I understand you've got antibiotic or no antibiotic, perhaps at a very macro level. But how do you see this clinically? So I understand the testimonials. I'm in the business, so I understand what you're trying to get to. But ultimately, how does it change patient care in the immediacy of what's happening? Because if I see a 2-year-old with severe fever, based on a single test result, if I'm to tell them to take antibiotic off, it seems the hardest decision the clinician is going to make. Without knowing a source, how are you going to get to that point of saying, look, I know everything about this, you don't need to treat it with antibiotic? How do you get to that decision point?
Okay, so again, this is a fundamental question. How do you create action? Because again, it's not about technology. It's not about multiplex PCRs, isothermal, CRISPR, ultrasensitive immunoassays. How do we actually create real actionability? And it's a really good question. So first of all, you talked about very severely ill child. So frankly, you don't need a solution for a very severely ill child. If that child has suspected meningitis, treat him.
I'm taking the same patient that you're taking, the same 2-year-old who is
okay.
ill.
We're talking about the, if I quote here, Rich Bacher, Professor Rich Bacher, we're talking about the moderately ill. I truly have a dilemma. Antibiotics, IV, so over there, look, frankly, most of these little buggers, they have a viral infection. Chicken soup. Back home, that's a solution. Don't think about only the technology. Let's think about what does the clinician needs to know in many of those cases. Can I send him home without antibiotics? Do I need to hook up to antibiotics IV or not? That's the question. If it's a viral infection, and in the winter, 85 to 90% of the cases is going to be a viral infection, that's it. Slam dunk. Now, absolutely, sometimes you have to go one step further. The example that Dr. Adi Klein was talking about in the text there, so here's a real concrete example where actually we didn't need to find out the bug. So she's calling, I was not there, but she was calling one of our medical directors, Dr. Manu, at Tuesday and telling her, look, we just had a case, 3-year-old child, a fever without source, admitted, antibiotics IV, but he started to look better. So, and we did the PCR adenovirus. So we wanted to release, and your score came 100. So you're creating basically a problem for me right now from a collegial perspective. But don't feel bad. Everybody messes up adenoviruses. They're notoriously known to CRP goes up, WBC goes up, everything goes up. So that was Tuesday, but just in case, she kept the child and did a CT, it was blank. On Thursday, she called me at 10 o'clock at night when they got the MRI result. It turned out to be a bacterial abscess in the spinal cord of the child, went to surgery, and she was basically telling me, well, we saved the life of the child. Now, to your question, yes, you want to hunt down the bacteria, but in this case, they wouldn't even hunt it down. They would actually send it back home. So is it important in some cases to go and hunt down the bacteria or the virus? Yes. But for the majority of the cases, often just getting this piece of the puzzle is good enough. And look, another layer on that, we have additional products that probe one level deeper. So this is just the beginning. We're not claiming to solve everything. It's just part of the problem, and we think it's an important part.
Understood. I believe we are.
Thank you.